Karen Lewandowski, I saw Carolyn in the Regional Perfusion meeting in Phoenix. She says, I got a great speaker for you. This young guy from the University of Pennsylvania, he's hot on the scene, and he's doing a great phase 2 trial looking at immunotherapy. And I said, we got to get him. We got Dr. Katz, and now we're going to bring this young rising superstar in to talk about his phase 2 trial. Today we're going to introduce, and I'm Thomas Karasik, who is at the University of Pennsylvania. He did his medical school at Columbia University and was recruited back, where he did his training at Penn, and then he has stayed on as a fellow at Penn, and he's now an attending at the University of Pennsylvania. And he is going to be talking about a clinical trial, which is really a phase 2 trial looking at nivolumab and ipilimumab for appendiceal tumors and mucinous colorectal tumors, and I'm really looking forward to hearing this next level of evidence in the treatment that we need to help our patients. Dr. Karasik.
Thank you, Dr. Bound, for the very kind introduction. Thanks for ACPMP, mouthful, for inviting me to speak. And today I'm going to talk about a trial that we just opened at Penn, a phase 2 study of immune therapy for mucinous colorectal and appendix cancers, and it's using 2 checkpoint inhibitors that are approved in other cancers, nivolumab and ipilimumab. So I don't think I need to give a whole lot of background to this audience who is very familiar with the disease, but we know, especially as medical oncologists, that peritoneal carcinomatosis from mucinous adenocarcinomas of the colon or the appendix really have very poor outcomes. Decreased responsiveness to chemotherapy and shortened survival compared to other sites of metastases. However, even in these high-grade tumors, there are a handful of patients who are long-term survivors who have generally gotten cytoreductive surgery and HIPEC. We know that that surgery is certainly not for all of the patients with the high-grade tumors because most of them will relapse and there can be significant morbidity. But there are some patients who do quite well. And for me, I think the genesis of this was realizing that perhaps some of those who do well, it's not so much the fact that all of their tumor cells have been eliminated, but that an immune response has been engendered that has been longer going. You really heard a great talk from Dr. Katz talking about the immune effects on these tumors, and our thought is that these mucinous tumors really do have antigens that are highly immunogenic and potentially can be targeted with immune therapy. And you just heard about some of the various other approaches, cellular approaches, antibodies, vaccines that are under development. But our goal was to use a lower hanging fruit, drugs that are already available, to try to engender an immune response. We know that PD-1 is important in this disease, and it seems to be more complicated than the usual mechanism we think of in hot tumors like melanoma, where T cells are all that matter. We do think it matters both T cells and B cells, and that there's probably a humoral response that's needed. And these drugs really haven't been looked at in mucinous tumors. These patients don't get involved in systemic therapy clinical trials, so we really don't know whether these checkpoint inhibitors work in the disease. You're going to hear in a much better talk, I'm sure, in the next few days from Dr. Allison about the drugs he developed. But in brief, I think simplistic terms, these immune checkpoint blockades take the brakes off of the immune system, and we're using 2 different immune checkpoint inhibitors in this trial. First, the anti-CTLA-4 antibody ipilimumab, and that seems to work mostly in the lymph node between the antigen presenting cell, the dendritic cell, and T cells to help then activate T cells who can travel to the tumor. And then in the tumor microenvironment, PD-1 and PD-L1 expressed by the tumor seems to then turn off the immune system. And so we're using nivolumab to block that inhibitory signal, allowing the T cells to proliferate in the tumor and kill tumor cells. The hypothesis, again, for this study is that cytoreductive surgery followed by HIPEC is really a form of immune therapy, and that the long-term survivors are those who have immune responses. As a medical oncologist, it's sort of anathema to think that a single dose of chemotherapy cures anybody, but we know that there are long-term survivors even with high-grade tumors, and there have been some mouse models showing that HIPEC is sort of a vaccination approach that subsequently rejects later tumor challenges. And so this is looking again at immune checkpoint blockade really as a signal finding study in these higher grade mucinous colorectal and appendiceal adenocarcinomas. Our goal, the primary objective of the study is to look at 6-month progression-free survival. You're going to hear from Dr. Siegelman in the next talk how difficult radiographic follow-up of these patients is. But we decided that this is probably a useful endpoint, both if they are stable clinically for 6 months, it probably means our therapy is effective. And that in cancers where this seems to work, getting to about 40% stable at 6 months seems to connote some survival benefit. And so that's our goal in this study. We'll look at response rate, although again, difficult to measure radiographically, duration of response, overall survival, and toxicity. And then really as important as the clinical outcomes in a small study like this is looking at measurements of whether we're doing what we think we're doing, activating the immune system, trying to figure out what really are the targets for these immune cells that are getting into the tumor, and also looking at circulating tumor materials to really try to complement what we're seeing radiographically to try to figure out who actually will respond to this and who we should be targeting in future studies. The endpoints and the statistics, as I said, our goal is 40%. We know that third-line chemotherapy just in colon cancer in general has very dismal outcomes with 6-month PFS of only about 17%. And so with 21 patients, we can really prove a significant benefit beyond that. And again, secondary objectives and probably as important, the exploratory objectives with some of our laboratory colleagues at Penn. This study is a fairly straightforward design. There is only one arm. Patients who are refractory to standard chemotherapy or have a contraindication or refusal of it will be treated with nivolumab at standard dosing, 480 milligrams every 4 weeks, and ipilimumab at a low dose to minimize some of the immune toxicities seen at higher doses, 1 mg per kg every 8 weeks. And they'll continue on until progressive disease withdrawal from the trial or up to 2 years. As I mentioned, we have a lot of correlative work, research going on with this trial. Patients will be getting biopsies both at baseline and at day 10. And that's based on work from melanoma showing actually with these checkpoint inhibitors, the immune response is actually much earlier than what we see, and that many trials sort of miss the immune response by doing it a month or two into therapy. So we're really looking early. And ideally, one of those is going to be via laparoscopy, where we can really get a lot of tissue and also then evaluate the patient if they respond well for future cytoreductive surgery. And they'll also get blood markers, both immune as well as circulating tumor materials at the beginning and then monthly throughout the trial. The key eligibility criteria, and certainly those in the audience, patient advocates, this is going to be a trial that's really limited to a select population as we really try to find the signal. This is for peritoneal-only disease, so not those with established metastases in the lung or the liver, really those who would be appropriate for cytoreductive surgery if they respond well. And it's for the higher grade tumors, the actual adenocarcinomas, mucinous, and both colon and appendiceal primaries are allowed. And those with suspected lower GI primaries that were treated as colon tumors, but a primary never was definitively identified, can also be considered for the trial. We're not targeting those where we already know checkpoint inhibitors work, the MSI high population. This is for the microsatellite stable. Those drugs already have FDA approval in the MSI high. And they should have received standard chemotherapy or have some reason why we shouldn't be given drugs that have more of a track record. Otherwise, the inclusion are standard for clinical trials. We do restrict that they cannot have had a bowel obstruction in the last 60 days because we know that immune therapy takes a little longer to work, and we want to give patients really a benefit of therapy. And this is my last slide. This is just showing, again, we're just getting up and running. We opened in February. We enrolled the first patient last month. We're looking for a total of 21 patients, and every single step along the way as I was designing this trial, I was told these patients are unicorns, but we really have every week getting multiple emails from people who are interested in this study. It is a select group, and so many who are potentially interested may not be eligible, but certainly if we are happy to hear from any patient who potentially would be eligible. These are the contact information for me as well as the study nurse and the nurse navigator. And so hopefully through forums like this, we can really prove that we can do trials, systemic therapy trials in this population and try to move the field forward. So that I'll stop. Hopefully got us back on time a little bit with a little bit of a shorter talk.